My name is Dr. Jonathan Miner. I'm an associate professor of medicine in the Division of Rheumatology at the University of Pennsylvania, Perelman School of Medicine. And today I'd like to give an update on our work on a disease called retinal vasculopathy with cerebral leukoencephalopathy, RVCL. I moved to the University of Pennsylvania, Penn, 13 months ago, and we have been completely dedicated to developing treatments for this disease. And we've made a lot of progress in a very short amount of time, and we're looking to make even faster progress in the very near future. So I want to tell you about it. So our goal is to develop personalized medicine for RVCL. So I arrived here at Penn 13 months ago, and we've already developed a gene therapy that works in cell culture conditions. So in other words, in cells growing in a dish in the lab, it can fully correct the disease-causing mutation in the TREX1 gene. And so there's still a lot of work to be done, but we are hoping to get prepared for clinical trials over the next couple of years so that we can then begin to test safety of gene-targeted therapy and correction of the TREX1 mutant gene in patients. But there is a long way to go before we get to that point. So we have a lot to do in a short amount of time. Importantly, RVCL is a disease that affects multiple organs, including the eye, the brain, the liver, and the kidney. And therefore, slightly different approaches are going to be necessary to treat the disease by targeting different organs. So we had a very nice meeting recently here at Penn with a lot of different scientists and physicians as part of our community. This is Dr. Rennie Ree. She's a rheumatology colleague of mine, a great clinical researcher, and she gave a very nice talk on how we're going to be able to share data from patients around the world with institutional review board approval, but the hope is to share anonymous data with anyone around the world who's interested in working on RVCL. The goal is to engage in team science where there are multiple groups actively pushing the field forward. We have an incredible group of collaborators. This is just one small snapshot of some of the people working with us here at Penn. This is Raul Coley, who is an expert in gene therapy, Sarah Cherry, who is an expert in the TREX1 gene and pathway and great at screening. She's also head of therapeutics at the New RNA Institute here at Penn. Dr. Ree, I already introduced. Jen Orthman Murphy is a neurologist here at Penn who has an undiagnosed rare disease clinic, neurological disease clinic. And she's been able to identify a genetic cause of disease in 25% of the patients who have been referred to her, which is remarkable. Jake Brenner is another physician scientist who is an expert in targeting therapies to different cell types in different parts of the body. And then me, of course, and then Martina Jimian, who is our new RVCL nurse navigator and who will be available to help any patient and family affected by this disease. I'll talk a little bit more at the end of the talk about some of the other highlights, but I think the key point from my perspective is that we are working in and among a group of scientists and physicians who are all sharing and facilitating progress that is directed toward therapy. This was one of the main reasons why I chose to come to Penn, was so that I could push therapies forward. And we've had great support, including from our chief of rheumatology, Peter Merkel, and also from numerous other scientists. Here pictured is Drew Weissman, who developed modified RNA technology, which has been revolutionary in medicine because it allows you to deliver RNA to specific cell types. And that may be something that we can, in fact, I'm certain it will be something that we will be able to use as we advance gene therapies for RVCL. So every day when I walk into campus, I walk past this painting, more than 100 years old. This is a class of Penn medical students observing a surgery, and they actually know the names of every single one of these students in this painting, which is remarkable. This is the old library, the Fisher Fine Arts Library on campus. There's a lot of history here. This was the first medical school in the country. The first hospital was here in Pennsylvania, in Philadelphia, and then in addition to that, it was the first university in America. So a lot of history. And as I walk past all of this history, I also think to the future. And I think about what we're going to be able to accomplish for patients with RVCL and for patients with a whole host of rare diseases for whom we ought to be able to personalize medicine to save their lives. This is a view inside the laboratory. This is our tissue culture room where we have actually worked to develop gene therapies as well as other therapies for patients with RVCL. There's still a long way to go. It's not ready for patients yet, but progress is happening faster than ever before. So what is RVCL? So this is a disease with many different names: RVCL-S, CRV, Hearn's, Chariot, and others. It stands for retinal vasculopathy with cerebral leukoencephalopathy. This was the first paper about the disease in 1988, Grand et al., first family pedigree. So the key issue in this disease is that there's a thickening of the blood vessel wall. And when you have a thickening of the blood vessel wall, it obstructs blood flow. When you lose blood flow, you get organ injury. So this happens in many different organs in RVCL. Onset of symptoms is usually between ages 35 and 55. As I mentioned, there's small blood vessel destruction. There is brain and eye disease in all patients with RVCL. There can be strokes and other neurological symptoms, kidney and liver disease in 100% of patients. There can be bone injury, osteonecrosis, hypothyroidism occurs. Sometimes there's gastrointestinal involvement. These things happen in a pretty reasonably high percentage of patients. So there are things to be aware of. And then premature death in all patients, but we intend to change that. So when you look at brain imaging, MRI imaging from patients with RVCL, you can see that some patients have large brain lesions and other patients have smaller brain lesions. And these lesions can change over time. They can become larger or smaller, and the brain can begin to atrophy. So this black space here is a ventricle. And as those ventricles enlarge, that means the brain is beginning to atrophy. And so this is what can lead to cognitive symptoms like memory problems or dementia. So this is a devastating disease, and we ought to be able to do something about it because we know the genetic cause of disease. So I want to talk a little bit about DNA, RNA, and proteins because I think this is important for understanding how we're going to develop a treatment for this disease. So the TREX1 gene is the code for the TREX1 protein. It's good to think about DNA and genes as blueprints for proteins, but the proteins are really the machinery that makes the body work. RNA is a copy of the DNA blueprint. So RNA is a kind of a temporary code for a protein. RNA is a very unstable molecule. It turns over rapidly. So when a gene is on, many copies of RNA are made so that you make many copies of the protein, but the protein is really the machinery that is doing the work inside the cell. And so if you have a mutation in DNA, you end up with having a mutation in RNA, and therefore a mutation affecting the protein. And a change in the protein sequence can result, or shape, can result in a change of the protein's function. And this can cause disease. So TREX1 has multiple functions. Its main function that's most well described in the biomedical research literature is to degrade DNA that's in the wrong part of the cell. I think that's the simplest way to think about it. But it's part of a network of proteins that interact with each other. And this can get very complicated, but I think the main thing to know is that TREX1 is an enzyme, which is a type of protein. And the type of enzyme is a DNase, which means an enzyme that will degrade DNA. So what does TREX1 normally do? Well, it has a function in DNA damage, so it can help fix DNA damage, and then it can also turn off inflammation by regulating the pathway that I had just shown on the previous slide. So mutations in TREX1 cause RVCL, and the mutations that cause the disease are all in this one region of the protein. And we now know of many mutations that cause this disease. In fact, we have been identifying new families almost every month from all around the world. So this disease truly affects people of all backgrounds and ethnicities from all countries, I believe. And as we sequence the genomes of more and more patients, it becomes more and more clear how widespread this disease is. So this is a picture of the TREX1 protein anchored in the membrane of a cell. And it actually functions in pairs normally. And you can see it has this domain or this region of the protein that crosses the membrane. Well, in a healthy person, there's two copies of TREX1, one from mom and one from dad. This is true for the gene, two copies of each, and two copies of the protein. Why are there two copies of every gene? Well, usually if something goes wrong with one copy of a gene, the other copy of the gene can compensate for the absence of the other copy. But in RVCL, that's not the case. So there's one mutation that eliminates this anchor into the membrane. And what ends up happening is mutant TREX1 ends up floating free, no longer anchored to the membrane. And so if you look at the fluorescence of TREX1 in this picture, where it's supposed to be, the protein, is just outside the nucleus. So the nucleus is in red. TREX1 is supposed to be anchored in a membrane that is outside the nucleus. But when you have a disease-causing TREX1 mutation, TREX1 ends up being found in the nucleus and throughout the cell, including in locations where it's not normally supposed to be. Somehow this leads to disease. We don't fully understand that yet, but we think we're getting closer to an understanding of it. So in this cartoon drawing of a cell, you can see that TREX1 is supposed to be in this part of a cell, but in RVCL, the TREX1 protein is located throughout the cell in different regions. So there was an interesting paper in 2008 where they found that there were certain DNA sequences called retroelements. So these are sequences that make up 46% of the human genome. They're not actual genes, but they are remnants of viral infections that occurred many, many, many years ago and then became part of the genome. And so TREX1 helps to keep these under control. So when you have TREX1, it's a small percentage of sequences. If you have no TREX1 at all, in other words, if the gene is completely deleted, which is not what happens in RVCL, but in situations where, say, an animal or a person has no functional TREX1, there's an expansion of these sequences, which we call endogenous retroelements. They're viral sequences. So it's 46% of the genome. So the key point here is that TREX1 plays an important role in regulating these. So these retroelements are kind of like a jumping gene. They, as we age, start to jump around the genome. And TREX1 can regulate that process. And so we think that this is playing a major role in RVCL, and we have evidence of this. And we've seen this, for example, when we try to, so there's certain viruses that will integrate their genetic material. They're called retroviruses. So they're related to retroelements that are part of our genome. So when we try to deliver a retrovirus into the genome of cells that have an RVCL causing mutation in TREX1, those viruses can't integrate or enter the genome very efficiently. So this is supportive of this idea that in RVCL, there's an issue handling retroelements. And in fact, we've looked at retroelements quite extensively, and we found that there are altered levels of retroelements in tissues from mice and in cells from mice. So it's pretty clear that that's part of what's going on. Now, in the human genome, there are 20,000 genes, and also all of these retroelements, which are separate from the genes. 3 billion bases, A's, T's, C's, and G's. And if one were to imagine the DNA from a single cell, imagine it the width of a spiral staircase, it would extend all the way to the moon. Now, RVCL can be caused by just a single extra letter out of 3 billion. It's something to think about. And so if we want to correct that, we have to be able to target that one mutant mutated base in the genome, which is not an easy thing to do. And we have to be able to correct it. And then more importantly, well, equally important is that we need to be able to deliver this gene editing machinery to specific parts of the body. We think we're getting a lot closer to doing that thanks to some wonderful collaborators that we have here at Penn. So there are many rare diseases, and there are over 300 million people affected by them. It's just there are so many rare diseases are common. It's just there's a very large number of diseases, and what I'm leading into is this idea that we need to have personalized medicines in order to help these patients with rare diseases because there's an unmet need. This is actually something that's gotten a lot of attention in the media lately. There was a Chicago Tribune article about RVCL recently, and then this Philadelphia Inquirer article. And as many of you probably know, that many patients with this disease have been told that their family has bad multiple sclerosis, or that they might have had brain tumors or other diseases like lupus. And of course, we know that those were not the correct diagnoses, and that the genetic information allows the correct diagnosis. So our field is moving very fast. 15 years ago, we knew of a limited number of families with RVCL, but now genome sequencing is becoming common, and we know of many families. We've been finding new families almost every month. We just found a family in India, for example. We're starting to find families in Eastern Europe. I need to update the map. And so as we find more families, it's just a reminder that the need is great and that we need to move as fast as we possibly can to develop an effective therapy, because this disease is devastating, and I believe we can do something to cure it. I don't want to get too excited about it, but I really am starting to believe that this is going to be a goal that we can achieve over the next decade, and hopefully a lot faster than that. But it's not going to be easy. There are going to be bumps in the road. Sometimes it's going to be 2 steps forward, 1 step back. And so everyone needs to be prepared for all possibilities. There are many families who still need to be found. And one of the ways we can find families is in our biobank. So when patients come to Penn, they get their genome sequenced, and we're able to search by gene, and then we're able to identify all the mutations and tie that to clinical information. So, of course, with IRB approval, but this is a remarkable capability, and it allows us to move things forward and identify new families. We've been working with the University of Michigan to develop small molecule inhibitors and to test them. We're hoping that these will become available, but again, there's still a lot of work to do, and we don't know yet whether this will work for RVCL. That's why we will need to do further testing in order to see, which drugs are most effective, which drugs might be safe, and we're still years away from being ready to do this. But we're closer with genome editing, and I think this is really exciting. So we have different ways to deliver the gene editing machinery. After a recent visit by one of the families to Penn, we had I got an email at 2 a.m. from Drew Weissman, who developed modified RNA, and Dr. Weissman said, "Send me the sequence of your genome editor. It's going to take 2 months to synthesize." And the collaborative nature of colleagues here who want to dive in and do the necessary work and help us, it has been incredibly inspiring and gives me a lot of hope. I don't want to give anyone false hope, but I think there's good reason to have hope because things are moving really, really fast now. So we're envisioning that Penn can be a center for the diagnosis and personalized treatment of rare rheumatic diseases, including diseases like RVCL. So we have a pipeline. We have model systems that include mice that have human genes in them. We have many different models of RVCL. We've made 3 new models since I've been at Penn, and they're models that I designed specifically with the goal of developing therapies. They're different from prior models in a lot of different ways. As I mentioned, we have all these therapies in development, and we have great partnerships with other institutions and with companies. We've also had support from the Colton Center. So the Coltons donated $60 million to Penn, and we were one of 4 pilot grant recipients, specifically to work on RVCL, which has been amazing additional support. And then finally, I want to mention Clayco Foundation and also the families and friends of patients with RVCL. As many of you probably know, every year in Chicago, there's this huge fundraiser called Illumination. I don't know how many people attend this. There are at least hundreds and hundreds of people there. And they raise a lot of funds that allow us to, and other investigators from around the world, not just at our university, but the support that we receive is enabling us to advance the development of treatments. If anyone has any questions about anything or wants to follow up or have a call with me, please feel free to reach out to me anytime. I will make myself available. We have a new RVCL nurse navigator named Martina Jiman, and she's also fully committed to helping and supporting families and patients with RVCL. We're also committed to working with other institutions. I've been communicating with Dr. Ford very directly at Washington University in St. Louis, and also with physicians locally for many patients all around the country. And I'm very happy to help provide support however I can, and want everyone to know that I'm committed to being available to them 24/7. We have an RVCL phone line that I keep with me, and I will be available to you anytime, even if it's the middle of the night. If something comes up, feel free to contact me and I'll help you and your family. Thank you.